Okay, great. Let's start. So good afternoon and welcome everyone. My name is Ludovica and I'm the outreach coordinator of the Future Vaccine Manufacturing Research Hub. So it is my great pleasure today to introduce you to the first webinar of the 2022 Platform Funding Webinar Series. Before starting, I would just like to remind everyone that this webinar is recorded and is public and will later be available on the VaxHub website as well as on the UCL Biochemical Engineering YouTube channel. So please, I would ask if you could keep your microphones and cameras off, but please do join the conversation via the chat. Be also aware that your contributions will be captured and your name may be visible on the screen and on the recording as well. And do not hesitate to email me if you want your contributions to be removed from the recording later on. This said, I think we can move on with a brief introduction to today's event. So just to give a little bit of a background, the VaxHub has been supporting an ambitious program of innovative research related to the challenges of developing, scaling up, and manufacturing vaccines of benefit to low and middle-income countries. So the platform funding webinar series that we're launching today and that will run throughout the next month was organized to showcase the great work achieved by a number of hub-funded feasibility studies. So back in 2020, several projects were advertised, applications reviewed, and then funds allocated as part of the Hub platform funding budget obtained from the Department of Health and Social Care. The Hub research was indeed financed by the government as part of the UK Vaccine Network, that is a UK aid program to develop vaccines for diseases with epidemic potential in low and middle income countries. So yes, today's webinar will present results from a collaborative feasibility study between the University of Leeds and the Centre for Process Innovation. This study aimed to determine the scalability of Pichia pastoris as a vaccine production platform for poliovirus virus-like particles, or VLPs. This webinar will address the need for new vaccines against polio and the recent work on VLP production and characterization, as well as immunogenicity. We will dedicate around 30 minutes to the presentation of this project and then allow a 20-minute Q&A session. So please feel free to post any question on the chat. So it is my great pleasure to introduce you now to our speakers, Professor Nicola Stonehouse and Dr. Lee Sherry from the University of Leeds. Nicola is a virologist interested in both the fundamental aspects of the viral life cycle and the development of novel vaccines, and she's leading a number of VLP-related projects. Our second speaker, Dr. Lee Sherry, is also a virologist interested in vaccine development and using heterologous expression systems such as yeast for VLP production. So welcome both, and thank you very much for being here today. I would also like to introduce Dr. Sara Planamente, molecular microbiologist from UCL, who will be chairing this webinar's Q&A session. So yes, once again, welcome everyone, and thank you for being here. Over to you, Nicola and Lee. Thank you.
Thank you very much. I'll organise sharing my screen.
Yeah, thank you.
Okay, hopefully you can see that okay.
Yes, that's perfect.
Great. So thank you very much for the kind introduction, and what we're intending to do over the course of the next half an hour or so is for me to introduce the problem. So introduce why we need a new vaccine against polio. What is polio and what's the problem that we're trying to address? And some of the work that we did sort of in an earlier part of our consortium work. And then Lee's going to take over and talk about much more recent data in terms of the work that we've done with the Hub and Scaleup and how that solves some of the problems that we had in the early part of the project. So as you heard in the introduction, we work on a variety of different viral problems. We work on fundamental aspects of the virus life cycle, also virus structure, and more increasingly so, vaccine development. And the group, based in Leeds, works in close collaboration with Dave Rowlands, who's Emeritus Professor of Virology. You can see his picture here. So on to polio. So polio is a disease which for many people actually causes very little problem at all. It's often asymptomatic, or it can cause quite a mild infection with quite a speedy recovery. However, for a small number of infections, paralytic disease can ensue, and this is due to motor neuron damage. So this can be temporary, or it can be permanent, and it also can be fatal. Polio is highly transmissible. It's transmitted through contaminated food and water. And so images such as this are a legacy of polio here in Nigeria. Now, we don't recognise really polio as much of a problem when we're considering Europe, the US. However, we still have legacy issues of polio here. And these two people contracted polio when they were children. This is Mary Berry, and she contracted polio in the UK. And she has some paralysis in one of her arms as a result. We also have an image here of Ade Adepian, who contracted polio in Nigeria much more recently, and is now known as one of our Paralympians and also TV presenter. So I think this illustrates, hopefully, what this disease can do. So before mass vaccination, this was the primary cause of disability worldwide. So disability was as a result of an infectious disease, which I think, really is quite a profound piece of information. Nowadays, polio is only wild polio is only found and endemic in Afghanistan and Pakistan. It's disease is caused by poliovirus. You can see this in this negative stain micrograph here. And the success of really reducing these cases of polio down quite dramatically is due to vaccination. And we've had vaccines for over 60 years. There are 2 vaccines which are currently in use, 2 broad categories of vaccine. We have an inactivated vaccine, so viral particles that have been chemically inactivated, and we also have a live attenuated vaccine, which is called OPV. So these are called IPV and OPV, and they're both have been used primarily as a mixture of all 3 polio serotypes. So these vaccines have really reduced the number of cases of paralytic polio dramatically over the last decades. And this has been a really concerted effort by the World Health Organization, by the Gates Foundation, by Rotary and others, with the aim to eradicate polio. And the data is looking really, really good. But the problem is there's the devil's in the detail in these very small number of cases that we've been seeing over the last 20 years. What are the problems? Well, one is that wherever we've got wild virus circulating, there will be outbreaks, and sometimes those outbreaks can spread to neighbouring countries. So this is a map from 10 years ago now, at a time when Nigeria, for example, still had a lot of cases of wild polio. More recently, we see a big reduction in the number of cases of wild polio. They're the little red dots that you can see on this image here, which is taken from the WHO website very recently. So this is data up to only a few weeks ago. So we're only seeing a few cases. You can see a total of 4. However, there's an awful lot of green dots on here and also some yellow dots, and those are vaccine-derived cases. And so because the the oral polio vaccine, which is very easy to administer, doesn't need special medical personnel, can be administered literally as a drop on the tongue. So it's a great vaccine, but it has a problem in that you can get recombination with other circulating coronaviruses, and you can also get mutations that mean that the vaccine itself can cause polio. And now we've been in a situation for the last few years where we're seeing a lot more cases of paralytic polio from the vaccine than we are actually from the virus, the wild virus itself. So what to be done about it? Well, there is a switch in many countries to using IPV, the inactivated vaccine. Not so easy to administer, but it doesn't have that problem. However, we still have a legacy issue, and that legacy are people who've been vaccinated with OPV and don't clear that infection that they may have developed. So these are called long-term poliovirus excretors, and they can basically put polio back into the water supply enough in order to basically set up an infection for others who aren't vaccinated. So because of these long-term poliovirus excretors, we have to ensure that polio vaccination continues, and that is, you know, a real challenge going forward when the number of cases are actually very low in order to try and keep vaccination going. So IPV is not going to be a great vaccine to do that, because as polio is eradicated, the inactivated vaccine will have to be made under more and more stringent conditions. So another solution is necessary, and we've known about that for a little while, and that's one of the reasons why we set up a consortium in order to develop a virus-free polio vaccine over 10 years ago now. And this has involved quite a lot of effort from quite a lot of people, and I can show you some images of some of those people. So at the top is us at the beginning, and the bottom is us quite a few years later. People have come and gone and joined the group, and this was the last time we were able to actually get together for a face-to-face meeting. So what were we trying to do? Well, we were trying to exploit the sort of the natural aspects of a poliovirus particle in order to produce an empty one. But polio is a complex virus. It's not just a case of producing one protein that you can then use that as an immunogen. The capsid here is made up of 3 different proteins, which you can see on the surface, and those 3 different proteins have to be produced from a polyprotein and cleaved by a specific protease. So it's more of a challenging problem. So this is also explained if we look at the genome. So the poliovirus genome here, towards the 5' end, encodes the structural proteins coloured here in green, VP0, VP3 and VP1. And VP0 can undergo further cleavage to produce VP4, which is internal, and VP2. And we know that because of studies, many studies on poliovirus assembly, that you can produce RNA-free particles, and indeed many coronaviruses produce RNA-free particles as part of the life cycle. And so these particles, in the absence of RNA encapsulation, don't have VP0 cleaved. So in lots of ways you could say that the virus has done the job for us. All we need to do is produce these RNA-free particles, and we've got our VLPs. Unfortunately, life was not so simple, and it's been known that there's been an issue with doing that for many, many years now. And that is because the particles without RNA and with an intact VP0 are not antigenically stable, and they convert from a native to a non-native antigenic form, even at really quite low temperatures. So that extra cleavage step of the VP0 stabilizes the particle and makes it stay in a native conformation. And so this is illustrated, I think, quite nicely from these structures from the University of Oxford, where we have native particles on the left and non-native expanded particles on the right. And I think you can see, hopefully, that the VLP has expanded, and that you can start to see a gap at the 5-fold axis right in the middle, sorry, at the 2-fold axis in the middle of the particle. And so these non-native particles don't elicit a protective immune response. So it's not a problem having some of them there, but you need the native particles in order to get that native wild type protective immune response. So what we set about was trying to stabilise the empty particles, and we started to do this in the context of working with virus and using evolutionary Darwinian approaches. So basically stressing samples of virus or in various different ways in order to select mutations. And as a result of this work, we have viruses that have a collection of mutations for each of the 3 polio types, polio 1, 2 and 3, that stabilise this native conformation. And so this is in the context of virus, using infectious clones, but then what we had to do, what we wanted to do, was to use recombinant expression. And so what this means is to take part of the viral genome that encodes the structural proteins, the P1 region as is called, and the viral protease, co-express those. We then get these cleavage events and assembly of a VLP. And we used a number of different systems. In Leeds, we focused on Pichia, and it's the Pichia work that we're going to talk and focus on today, although we have had success with baculovirus insect cell-based expression as well at the University of Oxford. So the vector of choice that we've focused on has been a Pichia pink vector, and using a number of different strategies, but the most successful strategy thus far, and the one that we're going to focus on today, is using this multisystronic approach, which means only one transformation step, which therefore is less metabolic stress on the yeast. We use a very lab-based purification protocol that Lee will talk a little bit more about at the current time, although we're moving forward beyond that. So we've been able to generate PV1, PV2, and PV3, all the 3 types VLPs in Pichia. They have a similar stability to samples of the current IPV vaccine, which is very promising. And I'm going to now focus on PV1 purely on the interests of time, but Lee will talk a little bit about some of the other polio types. So they also look good. They look like native particles. This is by negative stain EM, and this is a cryo-EM reconstruction showing that these VLPs do have native antigenicity and native structure, so basically look like virus. The proof of the pudding, though, really is what happens in terms of neutralizing antibodies and protection against disease. And there are 2 models which are used by our colleagues, part of the consortium at NIBSC. And these are a rat model where they look for the production of neutralizing antibodies, and a transgenic mouse model where they assess protection. And the rat model is what is used for assessing vaccine batch release for current polio vaccine. And so this is some data from PV1. So these are our Pichia produced VLPs, and we're getting a good level of protection. In fact, here it takes 2 doses of IPV, the current vaccine, to get protection, whereas here we can get protection with a single dose of our recombinant stabilised VLP. So that's really good news. The data for the rat tests when producing neutralizing antibodies was much more variable, and we would not necessarily get consistently good neutralizing antibody levels. Bearing in mind we were producing material very much on a lab scale with a lot of sort of variability in that production. And so that's something that has been improved more recently, but I'm going to let Lee tell you some of the results from that. So where we are at the moment is, as a summary, we've got stabilised polio capsids for all 3 serotypes. And we've selected those that have properties similar or better than virus and the current vaccine in terms of their stability, their structure, their antigenicity, and at least now we've got data to say their immunogenicity too. We are applying this approach to other picornaviruses such as enterovirus A71, which also can cause paralytic disease, and we have a separate project to do that. We are very involved at the moment in Scaleup, in looking at different purification strategies and starting to work with some companies in order to translate our results further. And we've been very fortunate to have 2 collaborations with the Vax-Hub. We've had a proof of concept work for cell-free expression in E. coli that I'm not going to talk about today because we've been asked to focus on the scale-up of the bioproduction process with CPI. And at that point, I'm going to hand over to Lee, who's going to talk about some of those results.
Great. So I think I can share my. Cool. Just need to get my presentation up. And hopefully everyone can see that.
Yep, that looks good to me.
Excellent. Yeah, so I'm going to talk a little bit about the scale-up process that we've done alongside CPI through funding with the Vax-Hub. So initially, we did an initial scale-up using PV1S stabilised VLPs, and that was initially funded by the Future Vaccine Manufacturing Research Hub. And we were using the AMBER 250 system. So that system was really good for us because it gave us a lot of opportunity to use a number of different conditions under bioreactor conditions to test whether different medias or whether different feeding types were able to improve our VLP production. So we then got the cell paste from CPI, we ferried it back to the University of Leeds, and then I started to do what is our very rudimentary lab-based purification to make our VLPs and then determine whether we were able to make particles under these conditions and whether or not they were also antigenic, as they previously were from our flask-based work. So Nick touched on this earlier, the purification process. So we had some trouble trying to work out how to do this at best, you know, when I started this project 5 years ago. But we have managed to come down on using the cell disruptor to bust through the yeast walls. So we do that, and that's temperature controlled because, as Nick mentioned, we do get an antigenic shift, particularly for wild type capsids at temperatures above 4 degrees Celsius. So we use the cell disruptor to bust open our cells. Then we do a couple of centrifugation steps to remove the insoluble material. And then this is followed by a chemical precipitation step. So in this instance, we use PEG 8000 to 8% volume per volume, and normally we do that overnight. This is then followed by a number of sucrose cushion and sucrose gradient autocentrifugation steps, in which point after our 15 to 45% sucrose gradient, we're able to fraction out each of those independent fractions and determine where we have viral proteins present, and then we can look at them through either through things like EM or by ELISA to determine the antigenicity of our particles. So Nick mentioned this earlier, and it's really quite important for a lot of the data I'm going to talk about, is the VLP antigenicity. So as you've got kind of a dramatized expansion of our particles. So what we're interested in is the native particles, which I'll refer to as D antigen, and we'll have what we call non-native, which are the expanded particle, and I'll refer to them as C. Previously, it's been shown if you were to immunize someone with C antigen particles, you don't get a long-term immune response, which is obviously not what you want from a vaccine. So what we do is to use try and get the native antigen, slightly less expanded particle, non-expanded, and what we found is that this produces a long-term immune response, protecting people against polio paralytic disease for a lifetime. So what was really good about this, we can detect both of these different antigen types by an ELISA. We have specific monoclonal antibodies that recognize either of these two shapes, which is really good, which means we can take our gradient fractions, and another postdoc on the project, Keith Graham, runs a lot of the antigenicity assays for us. And what we can see is if we go from the top of the gradient, which is fraction 4 on this ELISA, down to fraction 16 at the bottom of the gradient, we got the VLP sitting somewhere in the middle, which is really nice. We get a really good peak of D antigen, of native antigen for PD1S VLPs, and they are fraction between 6 and 8, peaking really high. And on this graph, we've got this pink dotted line across here, and this is BRP, or as our IPV control. So this is the current vaccine, and that is at 320 D antigen units per mil, which is actually equivalent to about 10 human vaccine doses. So as you can see here, even in just 1 mil fractions in fractions 6, 7, and 8, we've got equivalent to or above the amount of 10 human vaccine doses from a 200 mil culture. So we're really pleased with that level of production. And what was good about this particular prep, as we also saw, there was a lesser level of C antigen. As Nick mentioned, we can have C antigen present, that's really not a problem. But if we can try and put this ratio further towards the native antigenicity, that would always be better. And just on the right hand side, and Nick's already shown a great picture of this, the transmission electron microscopy negative stain, EM, that's been done, this one by Joe Snowden, and quite clearly they have this classical picornaviral capsid structure, icosahedral structure, which is what we're looking for when we're purifying our VLPs. So just kind of give a little bit of a summary of what we found from that first bit of seed funding, was that we were able to test two different media types. We were testing YPD or using methanols and juicer, and that is a very cheap, only takes three base ingredients to make that media. So we want to try that to see if we can try and make this vaccine as cheap as possible. And then we had a more complex media that was derived from the Pichia handbook on how to ferment and produce heterogeneous proteins within Pichia. Both these were done at 24 degrees. That was what was suggested in the handbook. And what we actually found is that we got a significant increase, almost twice as much D antigen being produced per in the peak, in terms of our peak D antigen doses from the YPD, from the cheaper media. And not only that, we were also able to see that we got improved D to C ratio in terms of our ELISA in our peak, which is really nice. We saw for both YPD and BMMY was a 7-to-1 and 5-to-1 ratio respectively. And this was improved over what we were seeing in the flask, where we only really got a 2.5-to-1 ratio. We think this was partly due to the change in the way that the methanol was being fed, because in the bioreactor this was being fed through a continuous flow method, whereas in our hands in the lab we can only do this through bolus feeding once every 24 hours. So I think this more continuous drip feed of methanol improved the rate at which protein is being both translated and folded to make these to make the particles. And part of the next set of funding that was set by VaxHub, we were looking to increase this expression temperature to 28 degrees in the 10-litre scale-ups, because this would then give us a comparison between what the expression temperature is in the lab versus what was recommended in the for fermentation via the handbook. So part of the VaxHub funding was to start with a 24 bioreactor Amber 250 experiment, and we did this for both our stabilised PV2 and our stabilised PV3 particles. So we then thought we'd switch up what we could test here, try and get as much information as possible from our bioreactor run. And as we ruled out BM-MY, BM-GY as being less productive than our YPD media, we used YPD versus minimal media. And this is because minimal media had been suggested as a way to select for better expressing clones of difficult to express proteins. And one of our serotypes, PB2 in particular, had shown to be a little bit of a problem child when we were trying to produce it in flasks. So we did this at those two medias, and we also used 3 different temperatures. So we knew what 24 degrees did for PD1, but we wanted to see if we had sort of stepwise increase by using 26, 28, and 30 degrees. And as before, we ferried the cell paste back to University of Leeds and used our assessment of whether the particles were being produced by gradients and then how antigenic these particles then were. So for the PB2 VLPs, actually in the Amber 250 run in the bioreactor, we saw really good levels of expression under YPD and YPM. And a spoiler for both PB2 and PB3, the minimal media produced nothing, which was really a little bit disappointing, but also quite good because it meant it reduced the number of variables that we had to then test as we try and optimize this for the larger scale up. So we had way above what is our pink positive line on each of these gradient antigenicity plots. That you can see it plots in front of you. The pink line represents what is the IPV for type 2, and that has 67 d-antigen units per mil. So each of the different serotypes require a different level of d-antigen for a human dose. For type 2, that's only 8 d-antigens. So as you can see here, we've got way above what is probably just under 10 human doses of PV2 in each of the different temperatures, which is really good. One thing that we can't do for PD2 is that we don't have a C antigen specific monoclonal antibody, which means we're then unable to determine what the ratio is here. But we were really pleased to see this level of D antigen being produced, particularly as in flask expression, we didn't see this level of production. If we move on to what the summary of those were, what you can see is there didn't really seem to be much of a difference in the level of d-antigen produced across the 3 different temperatures for PV2. Maybe a very slight increase by the time we got up to 30 degrees. We saw a very similar amount of d-antigen in our peak fractions. What we've got now in terms of our estimated total across the whole gradient. And in terms of our number of doses in our peak, we're between the 110 and 120, which is telling us that there didn't seem to be much of a difference between those temperatures. However, what we did see compared to our flask-based expression was there was approximately a 10-fold uplift, showing the vast potential for optimizing this process further as we go into the scale-up and optimize bioreactor fermenter-based expression. If I move on to PB3, what you can see here from these graphs is obviously in red, we got the C antigen, so we have got a monoclonal antibody that will recognize that. And we also have the black lines representing our D antigen. And what I think was quite interesting about this, again, we've got the pink line across, which is our standard. In this case, it's 288, which is again about 10 PB3 human doses. And again, across multiple gradient fractions, we can see we were producing far more than is in that final formulation, which is really, really good for us, even at temperatures of 26 all the way up to 30. However, what is interesting is that the ratio between the D and C, you can obviously see it changing visibly in front of you. Whilst it's not that different between the 26 and 28 degrees, you shift over and look at the 30-degree plot, you can see that red peak has dipped above that line, that pink positive control line, and you can see that the ratio between the black and the red is actually a lot lower, which is of interest. So if we look at the summary slide for that, you can see that actually at different temperatures, we focus on the final column here. You go at 26 and 28 because they're very similar. They're around that 2.5 to 1 ratio of D to C, which is good. Whereas as we went up to 30 degrees, despite the fact we may have got an increase in the amount of D antigen being produced, that ratio was less good. What we did see for PV3, which we didn't see for PV2 was a stepwise increase in the amount of D antigen being produced at each of those temperatures. And this was also a significant uplift on what we saw in our flask-based expression in the lab, between 2 and 4.5-fold increase. Therefore, moving forward, we decided to stick with 28 degrees because what we saw was that this gave us a good ratio of D to C antigen, but also was an increase over our 24 degrees of expression. So we thought, this was really promising results. We're really happy with how this was going, and luckily we had the funding to scale this up to 10-litre bioreactors, and we had the resources to be able to do 2 10-litre bioreactors each. And there is a wonderful picture. If you ever wondered what 10 litres of yeast look like, there you have it. It's appetizing. So we had the PV1 stabilised VLPs that we expressed at both 24 and 28 in these 10 litres to compare those 2 temperatures. As I mentioned before, PV2 stabilised VLPs were a little bit of a problem child, so we tried these under two separate mutants, both under 28 degrees and both under YPD, YPM. Whereas PD3, we did these at 28 degrees as well, but we did some mixed feed versus some methanol only feeding, just to see whether we could increase the level of antigen being produced. And what I've got next to save you going through all the same sort of graphs that we showed before is a summary table. And this kind of tells you how our scale up looks. And what you can see on here, we've got D antigen per 100 mil on the y-axis. On the x-axis, we have the different expressions that were done. So we've obviously got PV1, and we've got basing it based on our flask expression versus what we got from the 10-litre bioreactor, and just kind of show what level of increase we've had. And as you can see for PV1, see a phenomenal uplift of about 3.5-fold increase in terms of the antigen being produced per 100 mil, which is really, really good. Across from that, we have you can just barely see a bar for the PV2 original mutant, but when we did that bioreactor, that led to a 10-fold increase, which looks really, really good. And then there's a different mutant that we've been using that we're going to call SC5A, and that performed better in a flask. So that's why we want to try it alongside our other stabilized PV2 mutant in the bioreactor. And as you can see, that way outperforms in terms of de-antigen production, which is really nice. And we also saw the same sort of 2 to 3-fold increase for PV3 in the green at the end of the graph, which is really nice. And the other aspect that we were really pleased with was the increase in what looks like the particle purity under TEM. So you hopefully see under the flask express PV2SE6B, there are some correctly sized particles, but actually there's a lot of other particles in there, a lot of, whether that be misfolded, whether that be other host proteins in the process. Whereas when we did the bioreactor produced expression, what you got is a very nice clean background, and what is vast majority of particles in that image are 30 nanometer particles, as we'd expect. So we saw a really increased level of purity in terms of the our PV2 particles being produced by a bioreactor. So these VLPs that have been produced, or stabilized VLPs, PV1, 2, and 3. Here are some TEMs of those, and they all have that classical icosahedral particle structure that we were hoping for, which is really, really good. This EM was done by another postdoc on the project, Jeff Swanson. And all these VLPs have just been shipped to our collaborators at the University of Oxford, who are hopefully going to do some cryo wizardry and get us some high resolution structures of our VLPs produced in bioreactor to show they look really similar to those that we're producing from the flasks. So we're really, really happy with how that's turned out. And the last thing that Nick said I would touch on, we were interested in the immunogenicity here. So this is from the rat test, as Nick mentioned. This is the vaccine batch release assay. They don't have any poliovirus receptors, so they can't be challenged with virus. But what we're interested in is a number of neutralizing antibodies that the rats produce in response to whatever they've been vaccinated with. So in this instance, we got on the black circles, black line, that's our IPV, that's the current vaccine, and that's what we're aiming to hit. As you can see for PV2, that has a vast number of neutralizing antibodies, a single human dose. And then as you drop that down to half a dose, it still doesn't drop. Really, really good response, really strong response to the IPV type 2. In the blue squares, you can see actually we've got a very good neutralizing antibody response to the PV2 stabilised mutant at 1 dose, but it's not quite at IPV level. And this seems to drop fairly significantly as we drop down to half a human dose. However, one thing that we have been playing with, because often for recombinant vaccines, we do have to use adjuvants to try and boost that response. We have tried a adjuvant that is already used in a number of different childhood vaccines. And what we saw in the pink triangles is that response is equal to or above that of the current IPV vaccine, even down to where they start to cross over about 1/8 of a human dose, which we thought was really, really interesting, fantastic. We also see this across the other 2 serotypes, which is really, really good. So not only does this lead the way in terms of saying we can potentially pass this batch release assay in terms of immunogenicity of our particles, we also have the potential for dose sparing, which would actually increase the availability of vaccine doses, but also decrease the cost of vaccine doses. So therefore making it a really viable vaccine for lower middle income countries to produce of their own accord. So to summarise, just want to say that we've shown a very clear uplift in our expression level by going into the bioreactor. The AMBER 250 was invaluable in that it let us use a number of different conditions to test different medias, different temperatures and different ways of feeding to make sure we were trying to optimise this as well as we could before we got to the 10-litre scale. What we also saw was, when we got to the 10-litre scale up, we were seeing all serotypes increase significantly over flask-based expression, which is really good. And based on our conversations with people at WHO, it seems that we're at a very similar level to the amount poliovirus vaccine production levels, which is really, really good to hear. And hopefully in terms of our purification, if we can move that further away from our lab-based purifications into a more industrial scale friendly purification process, that may actually improve the amount of yield that we're getting. The TEM showed that the particles looked exactly how the coronavirus particles should look, so that was really good to know. And we're hopefully going to get high impact structures of those from the guys at Oxford doing the cryo-EM analysis. And finally, I ended on the PV2 stabilised VLPs, which show really good levels of immunogenicity when administered alongside adjuvant, which is really good. And we currently have a trivalent immunogenicity assay underway with our collaborators at NIBS, because as you all know, the current vaccine is given all 3 serotypes at once. So we're now doing the mixing each of the VLPs produced under the bioreactor conditions for PV1, 2 and 3, and hopefully going to see a similar response in the rats. So fingers crossed for good data from there. And I just want to finally just acknowledge members of the Team Polio within the Stonehouse group, so Nick and Dave as supervisors of the group, and also everyone who's kind of contributed to the polio project currently, Keith and Jess, Sue and Joe, and also everyone at CPI who've been invaluable for their expertise with working with the bioreactors and making it a smooth process for us to be able to produce this data. And that's me.
Great, thank you very much both. I would like now to invite Dr. Sarah Planamente to chair the Q&A session. So please, if you have any questions, just send them on the chat, and then Sarah will read them for you. Yeah.
Yes. Thank you. Maybe in the meantime, people upload or put their question in the chat. I have myself a question to you, Lee. I can see that you get, it's about the purity of your sample, because I can see that you have very pure and monodispersed particles. I can see that in your micrograph. And you know, I'm working with E. coli and purifying VLPs from E. coli. I'm using roughly the same method of you. I'm going through a cushion. I do a sucrose gradient, but I'm struggling with contamination, and in particular exosomes, so presence of vesicles. And you don't seem to have this problem. And I would like to know if this is more because it's a matter of using Pichia pastoris over E. coli that gives this advantage in terms of purity, or is there anything you do in your prep? Are you treating your sample with detergent? Are you doing something? So I'm very curious about it.
Yeah, sure. So there are two secrets here. One is that anything that goes onto the TEM for those sort of images normally go through 2 sucrose gradients to try and clean them up as much as possible. But the first thing is actually after we've done our sucrose cushion, we then resuspend in a particular buffer, which actually, to be honest, is affectionately known as Ellie's magic buffer, from Ellie Ehrenfield, Polio goddess, who told us how to use this. And this has a number of different detergents in it. So it has MP40 and sodium deoxycholate as part of it. The percentages off the top of my head, I can't quite remember, but I can forward it to you, and you can try to resuspend after your sucrose
There's
cushion.
somewhere that
Yeah.
can explain also. All right. Okay. So I can't see anything yet in the chat. People are a bit shy. Here we have a question from Adil. So can you explain again how you compare your interim to the standard IPV dose? You can see like in the chat.
Sorry, what I meant to say is the interim. So when you're doing your comparison, your gradients, you had your IPV standard going across there. I didn't quite understand the units or what that meant. So
I can take this if you want, Nick.
Yeah, I think you're best to take that.
So yeah, basically what we have, unformulated vaccine, which we use as a positive control for this. So we call it BRP, but it is the current IPV vaccine. And basically on our ELISA plates, we just run a number of different dilutions, and we know it's standardized to whatever doses it is per serotype. So for type 1, that'd be 320 D antigens. And basically what I'm showing you there is the raw OD numbers. So we can do proper analysis. You find like slopes of the curve to find out exactly how many D antigens there are. But initially, we're just trying to show that we have a lot of material there. So basically what that line is showing is what the maximum OD is on our ELISA, which is equivalent to 320 D antigen units per mil. And then we work backwards from there. Does that help?
Thanks, Lee. Yeah, no, I was just trying to figure out. It's like, okay, so this is a sort of a finite amount that you've got. So this is a bar that you've held, and you're just showing that actually you're able to, you're in your purification, your fractions, you're actually successfully going over that, which is really good news. Can I just ask a follow-up question, if that's while other people are thinking? So in a like-for-like purification, do you know how you compare to the IPV sort of benchmarks in terms of?
No, sorry, actually we really don't know, because actually I don't even know what the like-for-like purification would be for IPV. It seems to be that's quite a guarded secret. I think I don't really know how they do it. I don't know, maybe Nick has a better idea, but I know we've never done a comparison.
No, the only things that we've been told is that we're in the right kind of ballpark. And that's as far as we know. But I think there's still a lot of optimization that can be done regarding, yet more scale up and, as Lee alluded to, purification. So I think even with a sort of laboratory based scales, if we're on the right kind of amounts compared, from our sort of biomass of starting material to and units, that vaccine equivalent units, I think that's pretty promising for now. Yeah.
Yeah, I agree. Fantastic. Yeah. Well done, guys.
So I don't see any other questions in the chat. I might actually ask another one myself. How can you explain the huge difference in terms of like assembly of your VLP flask versus bioreactor? How can you explain that? Because you have quite a significant difference from, for instance, your PV2 that you don't have any antigenicity at all, and then you get like an increase. How can you explain flask versus, what are the main parameters that make the difference?
So for us, we think it's more to do with the greater control that we have initially. So in terms of the way that it's fed, so I alluded to this slightly in the talk, is that so when we're doing it by flask-based expression, it's basically the flasks shake away for 24 hours, and I come back and I add like a mil of methanol, and that's fine. But what that does is probably give a huge amount of induction material at once, and that can probably lead to a large drive in expression, and whether that leads to misfolding, misprocessing, particles not coming together, going into inclusion bodies or whatever. I'm not really sure. The other aspect that's led to this huge increase, I think, is the fact that the bioreactors can get to such a higher OD, means that we have many more yeast cells that are producing our protein of interest. I think it's about a 10-fold increase in OD as well. I think something like that. So that we saw compared to our flasks. So that made a big difference, I think, in the amount of materials being produced.
Okay, we have a question. So we have two questions actually. One from Vivienne. Is it not a problem to have animal origin peptone in your culture medium?
It's not something I've considered, to be honest. It's not something I've really thought about. No one's mentioned it as a problem as yet, but I'm sure that there must be an alternative. I think you can make bacterially derived peptone, I think.
Otherwise, we have a question from Sarah, actually a suggestion. There is a soy tone version available, which is animal-free. Actually,
thank you, Sarah.
All right. Okay. So I don't see any additional questions. So maybe I can go back to you, Ludovica.
Yes. Perfect. So
yeah, perfect.
That was great. Yeah, that's perfect. Thank you so much, Nicola and Lee. Was a great talk, very interesting. Thank you, Sarah, as well, for chairing the Q&A session, and thank you, everyone, for joining us. I would like to conclude this maybe just announcing the next webinar of this series, which is going to take place on the 9th of February, so in 2 weeks' time, same time. And I really hope to see you all there again. And that's it. I wish you a fantastic rest of your day. Thank you very much for joining us.
Thank you. Thank you, everybody.
Thank you. Bye, everyone. Thank you.
All right. Thank you.